Hi guys, welcome back. Let's pick it up. Chapter 17 or whatever chapter this is in the book that we're currently using for this class. We're going to talk about lipids, an introduction to lipids, fatty acids and waxes, what triacylglycerols are, the difference between fats and oils, reactions of those triacylglycerols, phospholipids, and then talk about steroids and cell membranes. All of this is really starting to get into the biochemistry portion of the GOB course, General Organic and Biological Chemistry. So let's see what this is all about. Lipids. Lipids are biomolecules that either contain fatty acids or a steroid nucleus. They are soluble in organic solvents but insoluble in water. And once we start to describe them, you will know that to be true. Lipids are defined by a physical property, not by the a particular functional group. It's more about how they're put together rather than one particular functional group. Everything else that we've talked about, alcohols, ethers, amides, carboxylic acids, all the other examples that we've talked about, amines, amides, all of these were particular functional groups. But we'll learn as we look that fatty acids and steroids aren't defined by a particular functional group. And we'll find that with a whole lot of biological molecules as we go. So because they're not limited to just a single functional group, but combinations, they have a variety of structures and functions, which, boy, if you're going to have a biological being, a biological form, life form, you're going to need lots of different structures and lots of different functions, right? Many, many non-polar carbon-carbon and carbon-hydrogen bonds, and very few polar bonds. So therefore, that's why they're highly insoluble in water. The Greek word lipos, which means fat, is why these molecules in general are called lipids. There are two general types of lipids that we have to be ready for. We already talked about these briefly, fatty acids and steroids. Four-member ring steroid molecule, and then there's the acid part and there's the fatty part. Okay. So let's first talk about the fatty acids. They are waxes. Fats and oils, which are also called triacylglycerols. Glycerol, phospholipids, and sphingomyelins. Okay, so big long words, big pieces of things stuck together. But as usual, when we're looking at these biological molecules, we think of the different parts, and then we think about the active parts. So relax. They're big words, but they don't mean quite so much, especially when we know how to break them down into the interesting parts and the sort of supporting parts. Now, the type of lipids that don't contain fatty acid chains, so if it's a fatty acid chain, it's a lipid, but if it doesn't, and it's still a lipid, then it's a steroid with those fused rings. Okay, there are the two types of lipids, fatty acids and steroids. Notice that steroids are all by themselves, and then of the fatty acids, we've got different types. Waxes, long-chain alcohol and fatty acid, triacylglycerols, so we've got glycerol and then fatty acids. So clearly, triacyl has something to do with the fatty acid. And if you remember, an acyl group is that, right? That's an acyl. Glycerophospholipids, so glycerol again. Got the fatty acids, but now we've got a phospholipid, phosphorus kind of a thing. And sphingolipids has a sphingosine, fatty acid, and then the phospho part. So one of the things that's sort of neat about this, that I like the way this looks, is because it really allows you to break things down into the important parts and the non-important parts, or sort of grouping things together so that what looks to be a little bit of a scary thing, all kinds of atoms and parts of molecules all over the place, really is nothing more than a box. You with me on this? It's just sort of a box. Fatty acid part, and then a long chain alcohol attached. We're going to look at this, and we sort of look at molecules close up, carbon double bonded to oxygens, and then we sort of back ourselves up, pull back, and look at the molecule as a whole. And then we even pull back even farther to look at parts of a cell or a membrane or a particular part of a cell. And then we pull back even farther and we start to look at an organ or part of a cell or an entire cell. And then we pull back even farther and look at an organism. So it's sort of the way you have to think about things. Biochemistry, you have to both think really, really close, even as close as 2 lone pairs on the oxygen and 2 lone pairs on this oxygen. And then you can pull all the way back to look at a cell or an organ or even an entire organism. Okay. So, let's see if we can figure out from a vocabulary way of thinking which lipids contain the alcohol glycerol, glycer, and the glycer, and then the glycer, and then the glycer. I think we got this, right? So which lipids contain the alcohol glycerol? Well, waxes don't, so D can't be right. Sphingolipids don't have a glycerol, so they can't. Steroids or waxes don't, so we would pick B. All right, so how are we going to categorize lipids? In general, we can think of lipids as one of 2 ways, either hydrolyzable, meaning can be cut and remove a water, or non-hydrolyzable lipids. They cannot be cleaved into smaller molecules, by aqueous water hydrolysis. Okay? So some can and some can't. Hydrolyzable and non-hydrolyzable. And we'll be able to identify them shortly. Okay, fatty acids. So when we look at this thing, what do we see? You actually have seen something a little bit like this before, I hope. I hope you recognize it. Long chain carboxylic acids there's the chain part, there's the carboxylic acid part with no branching, usually contain 10 to 20 carbon atoms, meaning nothing hanging off it like a chloro or a ethyl or something like that. That does not happen. It can be saturated or unsaturated, and we'll talk about saturated and unsaturated. Remember, well, let's do it real fast. Saturated means as many H's as possible. And unsaturated basically means double, or well, it's mostly going to be double bonds. Double bonds so that you don't have as many hydrogens as you could. And these things are insoluble in water. And there's no other functional group present. So this is the polar part, called the head. And this is the non-polar tail. What do these look like? Don't they look like soaps? Remember soaps? Remember how soaps looked? Same sort of deal. This is palmitic acid, in which there are 16 carbons with that one carboxylic acid at the end. Hydrophilic, right? Meaning likes water because it's polar. Hydrophobic means it's nonpolar, doesn't like water, doesn't dissolve. Okay. Hydrolyzable lipids are derived from fatty acids. And naturally occurring fatty acids, for some crazy reason, have an even number of carbon atoms. One wonders if the Marvel universe might find some strange way of inventing a superhero or a villain. I imagine the villain would have an odd number of carbon atoms in these fatty acids. I don't know, Fat Man or something like that. Be interesting. It's just very odd to me that naturally occurring fatty acids just happen to have an even number. It's just the way it is. Now, saturated, as we just talked about, fatty acids have no double bonds in their long hydrocarbon chains, where unsaturated fatty acids have one or more double bonds, and generally cis. Do you remember what cis means? Cis, same side. So chain, carbon, double bond, H, carbon bond, double bond, C. See, because these guys are cis, there's the chain. So it almost continues on like it's a straight chain. As the number of double bonds in the fatty acid increases, the melting point decreases. Why is that? Melting point decrease means less IMFs, right? Less strong IMFs. And what is that? That's due to the kinking of the chains. Anytime that everything lays flat, there's more interaction and the melting point is higher, but as soon as you start to kink the chain, they get to bump into each other more, or the kinking of the chain sort of crumples up. There's less interaction. And if there's less interaction, that means lower dispersion force. And if there's lower dispersion force, that means lower IMFs, which means you don't need to put as much energy in to melt the stuff. Important, be able to think through that rather than memorize it. Okay. Saturated fatty acids have only carbon-carbon single bonds. The molecules that fit closely together in a regular pattern allows London dispersion forces between the chains. And when that happens, they have higher melting points. There's just more places for contact, which makes them similar. And in particular, they're solids at room temperature. There's a bunch of examples. Lauric acid that comes from coconut, myristic acid from nutmeg, palmitic acid in palm oil, and stearic acid from animal fat. Big long chains. So 18 carbons, 16 carbons, 14 carbons, and 12. There they are. Now, unsaturated fatty acids have one or more carbon-carbon double bond. They're unsaturated, means they don't have as many possible hydrogens as you could possibly have. And that means they have kinks in those fatty acid chains. Even though the hydrogens are on the same side and this is cis, you can see there's definitely a kink in that chain. It doesn't go straight. Instead, it's sort of bent in a way. Okay? Because of that, they do not pack close together. Thus, they have fewer interactions, fewer attractions between the chains, which means it's easier to get it from the solid phase into the liquid phase. And so whenever we make unsaturated fatty acids, these are usually liquids at room temperature. Less IMFs. Oleic acid is a unsaturated fatty acid. You can see the bent nature of it. Unsaturated fatty acids with one are called monounsaturated for obvious reasons. One mono. Well, maybe you've heard of monounsaturated fatty acids. You're more likely to have heard of polyunsaturates. We used to preach that all the time in terms of um, intake of food and what's better for you. More than one, these are called polyunsaturates. Polyunsaturated fatty acids. Linoleic acid, linolenic acid, arachidonic acid. Okay, so if we were going to judge lowest melting point meaning easiest to melt, meaning the least IMFs, okay? And we've already decided that double bonds equal less IMFs. So anything that has a double bond would be easier to melt. This is saturated. I don't see any double bonds, do you? This has one. This has 1, 2, 3. And this has 1, 2. Okay. So I'm thinking, let's do them all. Let's do them all in order. The first thing we would look at to decide least IMFs in this case would be size, but they all have 16 carbons. So they're all the same size, basically. So if size, the number of carbons in the chain, doesn't work, then we look at polarity, but that's about equal, right? So then you have to look at unsaturation here. So saturated, which is C, would have the highest melting point, the greatest interaction between the chains. Strongest IMFs. And then we just go by 1, 2, 3, and 4. And then the question is, which one? Is it 1 or 2 or 3? And of course, more unsaturation, less intermolecular force, weakest IMF, therefore lowest melting point. And then D would come next, then A, then C. That's the trend. Nice test question. Be ready. Okay, omega fatty acids. You may have heard of these. Linoleic acid is called an omega-6 acid because of the position of the first carbon-carbon double bond in that long nonpolar chain. And we actually number not from the carbonyl-carbon, but from the other side, from the chain side. And clearly, that's the 6th carbon, so that's an omega-6 acid. Linolenic is called an omega-3 acid because of the position of its first unsaturation point. Its first double bond is at carbon 3. This is another example of how it looks all the way out. Linoleic acid, sixth carbon is where the first double bond comes, so we call that a omega-6 fatty acid. And linolenic, that's an omega-3 fatty acid. Omega-Cs are counted opposite the carbonyl polar head, and omega-6 fatty acids are found in plant oils, while omega-3 fatty acids are found in fish oils. You'll hear that on commercials sometimes when they'll talk up their vitamin that's full of omega-3 fatty acids. Okay, so let's draw some here. The condensed structural formulas for each of the fatty acids with 10 carbon atoms that follow. One is saturated. So saturated means no double bonds at all. Okay? So there's carbon, there's one carbon. So we're going to do 2, 3, 4, 5, 6, 7, 8, 9, 10. There it is. Now a monounsaturated, meaning one double bond, it starts at omega-3. So it's the same thing. One, two, three, 4, 5, 6, 7, 8, 9, 10. Okay, so CH3 for that first carbon, CH2 for that second carbon, and then what do we got? We got a CH double bonded to a CH, because the double bond happens at carbon 3. Got it? You don't want to have too many bonds on any carbon. So this carbon has a hydrogen and a CH2CH3, and then a double bond to another carbon that has a hydrogen, and the rest of the chain with the polar head group. 1, 2, 3, 4. 1, 2, 3, 4. Okay, that's nice. Monounsaturated omega-6. Let's draw it the other way. 1, 2, 3, 4, 5, CH double bond. So that's the sixth carbon. That's the seventh. 8, 9, 10 polar head. Nice job. Good job. So, omega-6 versus omega-3. We like omega-3. Why? Health-wise, they lower the tendency of blood platelets to stick together. So that lowers the risk of clotting. Lower the risk of heart disease. And that's good. And where do you find omega-3s? salmon, tuna, herring. Okay, all fish oils. Nice job. Okay. Waxes. Waxes are formed from saturated fatty acids and long chain, meaning high molecular weight, alcohols. The general structure of waxes are shown. You got your fatty acid with your carbonyl polar head, but instead of having the C double bond OH carboxylic acid group at the end, you make an alkoxy group with a long chain alcohol on the other side. So both of the R groups here are very long. That's what a wax is. So the general formation reaction of waxes is that dehydration, right, of a fatty acid and an alcohol. So we make a wax and a water. Okay, so they've got those long nonpolar carbon chains. Waxes are exceedingly hydrophobic, hence the waxiness, right? You throw a candle in water, it's not going to dissolve, it's not even going to get soft. It repels the water, it's hydrophobic. Therefore, they form protective coatings on birds' feathers. Did you know that birds' feathers had waxes on them? Absolutely. Sheep's wool as well. And they're also the coatings that prevent leaves of plants from losing water because they've got waxy coverings on the leaf. There's beeswax, which is myricil palmitate. Long hydrophobic region with a sort of a polar lump in the middle. Bunch of typical waxes. Beeswax, carnauba wax. They always use carnauba wax on cars. And jojoba wax, which is another one. They also make candles and soaps and other things out of the jojoba bush, and then they extract the wax from those. Okay, triacylglycerols, fats and oils. Triacylglycerols. Tri is a big hint. Or 3 esters. And you know what an ester is, right? C double bond O R O R. That's the general format, functional group of an ester. There are 3 esters formed from glycerols and 3 molecules of fatty acids. All right, so it's major energy storage, triacylglycerols in animals. There's a polar bear, which basically eats like crazy during the winter, stores up its fat energy, and then doesn't eat very much for long periods of times, but lives off the fat storage. There's the glycerol. There are the 3 fatty acids that are attached, and these are the ester bonds. Let's take a look at what that looks like, not from the far view, but from the reaction point of view with the atoms and molecules. The general reaction is we take a glycerol, which is a 3-hydroxyl group alcohol, and then we react it with fatty acids. And I hope you see what happens here. It's a dehydration reaction again, in which water comes off, and the fatty acids stick on to make the ester bonds. Okay. The hydroxyl groups of the glycerols react with the carboxyl groups of the fatty acids, and you make yourself a triacylglycerol. So simple triacylglycerols have 3 identical fatty acid side chains. Same stuff, same stuff, same stuff. But mixed triacylglycerols have 2 or 3 different. Like you can have a stearic acid, oleic acid, and a palmitic acid. That's 3 different fatty acid chains that get attached to the glycerol molecule. And of course, we have saturated and unsaturated. Saturated triacylglycerols contain only saturated fatty acids. That's what animal fats are made up of. These are solids at room temperature, right? Think about it. Steak fat. Could look at it that way. Steak fat is a solid, right? And can you liquidize it? Yeah, go ahead, cook some ground beef. All the fats are ground up into the meat, and at room temperature, it's a solid. It's not like you see the meat on the shelf swimming in like a oil or something. Ugh. It's just solid, but all you got to do is heat it up, and what happens? You make all that grease, right? The meat cooks, the grease goes from solid to liquid. So there's a saturated triglycerol. Look at that. No kinking, no double bonds. Everything's sort of laying right up against everything else. Now, unsaturated contain at least 1 unsaturated fatty acid. And most vegetables oils are unsaturated triacylglycerols. And as you know, most vegetable oils, corn oil, safflower oil, canola oil, vegetable oil, they're liquids at room temperature. You can buy them in bottles at the grocery store. So obviously, if they have liquid at room temperature, they have lower melting points than the saturated, for all the same reasons we said before. Now, there are different kinds of unsaturated. There's mono, 1, and there's poly, many. And increasing the number of double bonds decreases the melting point. Decreases the melting point. Increases the kinking, which means lower IMFs, less intermolecular forces, which makes it even more liquid-like, easier to get into the liquid phase. And that's a picture of an unsaturated glycerol. Look at that. That's definitely sticking way out there, such that if there were more of them coming along, they wouldn't be able to lay next to and rub up against nearly as easily. Okay. Triglycerols, general features. Fats have higher melting point. They are solids at room temperature. They're derived from fatty acids with very few double bonds, whereas oils have lower melting points. They're liquids at room temperature, and they're derived from fatty acids having larger number of double bonds. So just keep that in mind. That's an important thing to think of. Okay, so always remember that the physical things that you can see are obvious, right? Fats on the side of a steak are solid. That means that the intermolecular forces must be higher, which means they get to rub up against each other more, which means it's saturated with no double bonds. Oils are liquidy, which means they're easily able to get up into the liquid state, which means the intermolecular forces are less, which means they must not be able to rub against each other as easily. The dispersion forces are less, and that happens with kinking, and that happens with unsaturation and double bond formation. Okay. Triacylglycerols. Okay, foods rich in saturated triacylglycerols. All that stuff. And this is sort of interesting. Remember, you're not eating cholesterol. We'll talk about this in a bit, but these substances end up making blood cholesterol. And look, cholesterol isn't bad. We've learned a few things over the years. It increases blood cholesterol, and that's not good. So this food isn't as really all that good for you, right? But it's not because you're eating cholesterol. It's because you're eating this stuff that ends up making cholesterol into the blood. Now, these are polyunsaturates, which are good. These decrease blood cholesterol level just based on how they're synthesized in the body. And then trans triacylglycerols are not good. We used to think that margarine was so much better than butter. We're finding out that's not the case, and you'd be much better off using this kind of stuff. So you want to try to reduce these and increase these in your diet. Okay. Now let's start with some chemical reactions of these. Just talked a little bit about what one might be. So let's roll with those. Triacylglycerols undergo 3 basic reactions. They're similar to what we've already looked at in alkenes and esters. So what we're doing clearly is what we talked about earlier. Biochemistry is little spots on great big molecules where we find the specific functional groups and do reactions at those individual spots. Okay? So there's hydrogenation, hydrolysis, and saponification. You should know what hydrogenation is. You should know what hydrolysis is. And we'll talk about saponification. I think we talked a little bit about it. You'll see it. Hydrogenation means add hydrogen and take the double bond and turn it into an alkane. Okay? Or take an unsaturated fat, which has double bonds, and turn it into a saturated fat with single bonds by adding H onto a double bond. Okay? Hydrolysis means cutting water. So what do? We have an ester, and in the presence of acid and heat, what we do is we chop the ester apart and make the carboxylic acid and the alcohol. In the triacylglycerol, we need 3 waters, and we separate out the fatty acid part from the glycerol molecule. And in saponification, remember this one was in acid, and this one, saponification, is in base. have an ester, react it with base, and what do we end up making? Not the carboxylic acid from the acid, but the salt of the carboxylic acid, the carboxylate ionic compound, and then the alcohol. And what are we going to do over here? We're going to take the triacylglycerol and the sodium hydroxide. We're going to make the alcohol, the glycerol, same as this, and we're going to do not the fatty acid, but the sodium salt of the fatty acid. Same stuff, just with bigger molecules, but with the reaction at the exact same functional group that we saw before. Cool. So in hydrogenation, not hydration, but hydrogenation, double bonds react with H2 in the presence of nickel or another metal catalyst. And we turn the double bonds into single bonds. Okay, now whenever we're going to talk about hydrolysis, triacylglycerols are hydrolyzed with water in the presence of acid or base or enzymes whenever this happens in the body. Okay, now the water adds to the ester bonds. We're releasing a glycerol, the alcohol, and all three hydroxyl groups come off, right? Because that looks an awful lot like glycerol already. We just take hydrogen stick them on here, and then we take these three OHs and we stick them on here, here, and here to make the carboxylic acid. Okay? We've seen that before. We've seen it. We make the three fatty acids. Remember, if you're in acid, you'll make the fatty acid. And saponification, soap synthesis. What's the first rule of Fight Club? All right. Soaps are metal salts. And metal salts means ionic compounds. Fatty acids prepared by basic hydrolysis. You need base, you need lye, or some strong base to make soap. So saponification of a triacylglycerol. Okay, so there's sodium stearate. Stearate, not stearic acid, but stearate. And this is a very common soap material. So you didn't make the OH, you made the sodium salt, because this is a minus charge. And then that's the plus charge. And that's the basic soap. Polar head, non-polar tail. So how does this work in particular? 3 sodium hydroxides in the presence of water come along, and what are we going to do? We're going to chop off this again. We're going to make the glycerol, so it got an H. And then what do we, it almost as if it looks sort of like, well, no, I don't really want to do that. And then what comes on, instead of making, right, because in base, you're not going to make the acid, you're going to make the salt. So it's an O minus and A plus, and you're going to make 3 of them. And sodium oleate, man, that's soap. Okay, now let's talk about phospholipids. Phospho contains a phosphorus atom. Okay, there's 2 common types. Glycerophospholipids and sphingomyelins. Big long words, don't be freaked out. It's more important that you recognize what we just did, the glycerol and the 3 fatty acids attached. And they're very similar. We got sort of a glycerol foundation or structure. And here, a glycerophospholipid has 2 fatty acids, lipid-like, and then we've also got the phosphorus compound hanging off it as well. And then instead of a glycerol structure, we have a sphingosine with a fatty acid, and the same sort of, I think that needs to have a P in there, sphingomyelin. Okay, so let's explain what these are and their importance. Glycerophospholipids are the main component of most cell membranes. Gee, I guess that's important, isn't it? There you go. So now we're getting even deeper into biochemistry. This is why we're studying this. Structurally, they resemble a triacylglycerol, just like we just talked about, except instead of having 3 fatty acids, we have 2 fatty acids and a phosphodiester bonded to an alcohol. Okay, phosphodiester looks sort of like an ester, OR, OR, but it's got 2 of them, bonded to an alcohol. All right, so let's look at what that looks like in a little bit more closer view. 2 main types of glycerophospholipids. One is called cephalin, and there it is. And the other one is called lecithin or lecithin. And there it is. And this is derived from ethanolamine. It gets chunked onto what almost looks like a PO4 negative 3-ish phosphate. And this is similar. Choline, that's actually something that we'll run into later on, amino acid part. Okay, they are abundant, these are in brain and nerve tissues, and we find them when we eat egg yolk, wheat germ, and yeast. Okay, so for glycerophospholipids, how do we think about them? Well, the 2 fatty acid side chains form 2 nonpolar tails, just like we would expect them to, that lie parallel to each other, which makes them soluble in nonpolar organic substances. Okay, but the phosphodiester end is charged. Or in other words, it's got sort of a polar head on that part, and that makes it strongly attracted to water. So that almost looks soap-like, right? The polar head. This is all the polar head part. And then we got the nonpolar tail. Might be straight. Here it's branched. Okay, so why is that important? Well, where do we start to look at a cell membrane? You'll see what we mean by that, why it's important. All right, but before we do that, let's talk about sphingomyelins. Sphingomyelins are phospholipids that don't contain the glycerol backbone, but they have a sphingosine backbone instead. This is sphingosine. So there's some similarities to the glycerol. Okay, some similarities, but there's also definite differences. Okay, so it's called an 18-carbon unsaturated amino alcohol. Look, this is just how nature or God or whoever it is that made it. So this is just what real life is all about. So let's keep looking. Sphingomyelins do not contain an ester. Their single fatty acid is bonded to the backbone by an amide bond. You know what an amide, right? R, C, double bond, O, N. So let's see it. Let's find that amide bond. Let's take a look at it. Here's a good example of a sphingomyelin. There's the long chain. And there, C double bond O R N. That's the amide right here. That's the amide that's formed with this long oleic acid fatty acid chain. There's the phosphodiester part with the amino acid. Okay, and why is this one important? The myelin sheath, which is the coating that surrounds nerve cells, is rich in sphingomyelins. This is a big deal. Why is it a big deal? The myelin sheath is the material that surrounds the nerve cells in these little sort of pockets. And they're very, very good at instant, quick nerve cell information transfer. That allows us to have quick twitch movement. When it's a healthy nerve cell, that's like sort of, do you remember when you played Chutes and Ladders as a kid and you'd hit the one and you could shoot fast over to the next level by just running right down the ladder? That's sort of how this works with these myelin sheaths, okay? And that's how the proper functioning of the nervous system works. However, as you can see, it increases the speed that the impulses travel and let you do things very quickly. But when you have scarred myelin, in multiple places, you end up with a disease called multiple sclerosis. And multiple sclerosis slows down or blocks the messages because the signal doesn't go zip, it kind of goes, and therefore you can't do the things you want to do. It slows down your responses, and you end up with a loss of eyesight, speech, mobility. It's a degenerative disease, and it's devastating. Multiple sclerosis has everything to do with the sphingomyelin. Okay, one final topic in general on lipids. We talked about fatty acids. Now let's talk about steroids. Okay, steroids are a group of lipids, have several fused rings. Every steroid skeleton looks just like this. It has 3 cyclohexane rings, A, B, and C, 1 cyclopentane ring, 1, 2, 3, 4, 5, which is D, no fatty acids at all. It's got those 2 methyls hanging off right there and right there. And steroids include both hormones. Hormones are a steroid, and cholesterol is also a steroid. Okay? Good. Okay, so it should be obvious whenever you see a steroid. It's that 4-member fused ring thing. And fatty acids is that long chain with the tail. Should be pretty easy to at least identify what is what. Okay. All right, we went through this process. Now, cholesterol, which is definitely the most prominent steroid, for lots of different reasons, is synthesized in the liver and found in almost all body tissues. Cholesterol is not bad, guys. Cholesterol is essential. If we didn't have cholesterol, basically we would just be sort of like Jell-O, because cholesterol has an awful lot to do with the structural integrity of cells. So that's what cholesterol looks like. And we get it from eating milk, butter, eggs, and meat. Okay, so what is what? Okay, what would you call A? Well, that's nothing more than a hydroxyl group. What would you call B? That's a methyl group. That's all that is, right? What's C? C is what is known as the steroid nucleus. Right? And then what's D? Well, D is nothing more than an alkyl chain. And that, my friends, is what all assembles and puts together and makes cholesterol. Cholesterol in the body is needed, okay, for cell membranes, brain and nerve tissue, steroid hormones, vitamin D. We need it. And the typical diet includes way too much. And whenever we end up making too much cholesterol, there's diabetes that are involved and lots of cancers, in addition to clogging the arteries. because cholesterol is not soluble in blood. It travels as little hunks and little pieces, and it gathers on the sides. These spots are the only places where the blood can get through on this particular artery. The light, okay? This is a big wide open cavity, and here it has to slide through all this stuff. Slows down, that means clotting, that means heart attack. Dangerous. Elevated levels of cholesterol in the bloodstream lead to bad stuff, mostly coronary artery disease. Because cholesterol is insoluble in the blood, it travels as a solid. It's transported by lipoproteins. Now remember, it has to go. We need to be able to transport the cholesterol from the liver where it's synthesized to the cells where it needs to go to make new cells. So it has to be done. Now we have low density lipoproteins, LDLs. They transport cholesterol from the liver to the tissues. And the high density lipoproteins, HDLs, transport the cholesterol from the tissues back to the liver. So what's a lipoprotein? A lipoprotein or a lipoprotein, they're spherical structures that transport hundreds of nonpolar lipids and cholesterol esters through the aqueous body fluids. Okay, they're like little micelles, aren't they? Lipoproteins combine lipids with proteins and phospholipids. They're soluble in water because of that polar hydrophilic outer coating, if you will. All right. To the outside, it says, hey, this is great. Let's dissolve. Inside is all the non-polar stuff. But it's covered by the polar. And so, as a result, it's soluble. So the LDLs, what do? They deposit the cholesterol on the walls of the arteries because they can't hold it when they carry more than is needed to form the cell membranes. And that's not good. This forms plaque, and that's not good. So the LDLs are called the bad cholesterol. If we have too much of that, it forms the plaque. HDLs reduce the level of cholesterol in the bloodstream by bringing the excess back. So this is the good cholesterol. It finds the bad cholesterol, grabs it, and takes it back to the liver. Okay, so let's talk about hormones. Hormones is a molecule that is synthesized in one part of an organism and then elicits a response at a different site. It's made someplace, and then whenever it gets squirted out or carried over there, something else happens, some response at a different area. Steroid hormones are the chemical messengers in the cells. They're produced by reactions with cholesterol. Cholesterol is the building block for all steroid or hormones. They are in two important classes, which are the sex hormones and the adrenocortical hormones. adrenocortical hormones. Okay, so what's the difference between an adrenocortical hormone and a sex hormone? Well, female sex hormones, probably know about these, are estrogens and progestins. Whereas the male sex hormones are called androgens. Okay, so let's dig into these a little bit. The estrogens, estradiol, and estrone, there's estrone and there's estradiol, control the development of secondary sex characteristics, regulate the menstrual cycle, and they get made in the ovaries. And you can see the steroid backbone. Very good. Now, progestin, that's called progesterone, I should say, progesterone. Think about the gestation period. It's called the pregnancy hormone. It's responsible for the preparation of the uterus so that the fertilized egg can be implanted on the uterus. There it is. That's the baby maker, or one of the things that helps. Now, as far as the male sex hormones, the androgens, testosterone and androsterone, now we call it testosterone. Do we call it androsterone? I don't know, maybe we do. I've always heard it as andro, so androsterone are androgens that are made in the testes, and they control the development of the secondary sex characteristics in males. There's what testosterone looks like, and there's androsterone. Again, the steroid backbone. Good. Now, adrenocorticosteroids are steroid hormones that are produced by the adrenal glands that lay on top of the kidneys. Adrenocorticosteroids, for examples, are cortisone, aldosterone, cortisol, and prednisone. And you've heard of some of these. Cortisone shot, cortisone treatment, athletes get those all the time. Prednisone is something that you get if you have a really bad bee sting. They'll hit you with a steroid shot of prednisone to try to stop the allergic response. And again, what is that? It is a hormone that is released in the body that causes an effect somewhere else. Cortisone injected in the body lowers inflammation in a part of the knee or something like that. Okay? Now let's talk about cell membranes. We'll go back to those sphingomyelins in just a second. All right, cell membranes. The basic unit of any living organism is the cell. And it's the cell membrane that's sort of a genius design, because it separates the content of a cell from the external fluids. If we didn't have that, basically it would all look like Jell-O. It acts as a barrier to stop the passage of stuff into or out of the cell. So you want to make the environment different from inside and outside the cell, and you need a cell membrane to do that. But you need stuff to go into the cell sometimes. It allows nutrients in and waste out, making the membranes selectively permeable. Meaning permeable. Meaning When you want stuff to come in, it can come in. And when you want stuff to go out, it can go out. And if you want stuff to stay in, it stays in. If you want stuff to stay out, it stays out. Selective in its permeability. Okay, so this is the generalized structure of the cell brain. There it is. There's the phospholipids. Hydrophilic polar head and 2 hydrophilic non-polar tails. That's that sphingomyelin that we were talking about. And it's a little bit soap-like, right? Here's the hydrophobic interior. Non-polar with non-polar, like dissolves like. So they like being in there. And then we've got the polar heads out here. This is why this is called the lipid bilayer. Okay? So when phospholipids are mixed with water, they assemble in a lipid bilayer. That's what all cell structures are like. 2 rows of phospholipids with the non-polar tails facing each other and the polar heads in the outer and inner. The polar heads orient on the outside to interact with the polar water molecules that are out in the cell environment, and the non-polar tails orient on the inside to avoid all that polarity of the water and the fluids around the cell. Okay. Now, proteins, carbohydrates, and cholesterol molecules are embedded in the lipid bilayer. Then you have peripheral proteins. Are embedded within the membrane and extend outward only on one side. So they extend out into the environment outside the cell. Those are at the periphery on the outside. Your peripheral vision is on the outsides, right? And then we've got the difference, the opposite, are internal proteins that extend through the entire bilayer. And they go right through the inside. And there sometimes we have carbohydrates get attached to the exterior surface of the cell as a way of communicating with hormones and neurotransmitters and communicate in chemical reactions. Everything is a biochemical reaction when interacting with the cell. But that's still a communication. And the lipid bilayer has unsaturated fatty acids to make the cell membrane more fluid-like rather than rigid. Because unsaturated means lower IMF. Why do you think we talked about this all the time? And if it's lower IMF, it's easier to melt. And while we're not melting anything, it's definitely making it more like a liquid than like a rigid solid. All right? So let's take maybe a closer look at this. There's a cell membrane. So what do we got? That glycerophospholipid bilayer, polar heads, non-polar tails hung together. There's a carbohydrate that's just sticking out. There are proteins that go the whole way through. That's an internal or just a peripheral protein. We've got cholesterol also in the through the cell membrane, hydrophilic, hydrophobic region. Inside, we've got the nucleus and then cytoplasm. That's an amazing thing, but all of it has to do with what we're talking about right now, these biomolecules. So what can get through, what can't get through, how does it work? Transport across a cell membrane. Very small molecules like oxygen and carbon dioxide can diffuse, traveling from higher to lower concentration. Think of drops of food coloring. Right? If we drop food coloring into a glass of water, bloop, right? That's where it starts, and then it slowly, you don't have to do anything, starts to spread out. It diffuses throughout everything, spreading out through all the water molecules, and if you wait, I don't know, an hour or so, maybe even less time, what is it? It's evenly spread out. You don't have to do anything. So these automatically go. Okay. Now, larger polar molecules need facilitated transport to cross efficiently. In other words, you got to help it. Needs help. Oxygen and carbon dioxide, they can just go anytime they want, but we got to make this happen for larger polar molecules. And ions travel through protein channels. Sometimes ions have to go, and they can just go right through that hole, sort of, that the protein channel has. Some ions, other ions, they move against the concentration gradient. Now that's not easy to do. They're basically going in through the outdoor, if you will. And that requires energy input, and it's called active transport. It's a little bit different than facilitated transport. And we'll get into this in a bit more detail in the future. So, diffusion, also called simple or passive transport, moves the particle from a high concentration to a low concentration, right? Whenever we drip the food coloring is high concentration and the water is low concentration, and the food coloring molecules spread out from the high concentration of the initial drip into the entire glass of water so that it spreads out, moving from high to low concentration. Then there's facilitated, which uses protein channels to increase the rate, okay, finds a way to help it push through, sort of grabs it and yanks it through. And then active transport, move the ions from low concentration to high concentration. Energy is required to make that happen. All righty.